And depending on how far we get, what I'll do when I get back to my office, I will send a set of questions that you can use to kind of, go over things, to get ready for the exam. And then as you're going through those, if there are any questions, please let me know before, end of the week. That might be the most productive thing to do, actually. So, yeah, so when I get back, I'll send that. But, so we have a number of things to do today. We want to kind of finish up talking about endodontic lesions, how those get caused, and some of the microorganisms, bacteria that we find associated with lesions. And then we want to look at systemic infections, how we get bacteria out of the mouth, and into the blood and to places like the heart and so on. And then we'll look at, gram-negative organisms, how they cause disease, and in particular, we'll look at Salmonella, we'll look at, E. coli strains, and then we'll look at the cases. And so, the way we're going to approach the cases, normally, you would each have a copy of the cases and read them, and we'll do a discussion. But because of time, okay, we can't do those kind of things. So we're just going to highlight certain things about these particular infections, and most importantly, these are the kind of infections that generally dentists in practice may never see. But depending on where they practice, and depending on where they go, depending on what kind of research and programs they're involved with, they definitely will come in contact with these conditions. And the other reason why I like to have us look at those, and whenever I do the class, I bring these up, is that sometimes some of these conditions, when we read about them, we think of them as being far out over there somewhere, and no need to really think about it. It's not really something that you're going to encounter. But what's important to note is that right here, in the U.S., depending on what's going on with individuals, they may actually have things like like luma. And so when that presentation is made, how do you know what it is that you're looking at? And so the two examples that we have of the case studies, are just that. We have one case, here in Atlanta, Georgia, and then we have another case, from overseas. So when we get to that point, we'll talk about them. But so we have started to look at some of the things that happen, what we have in the in endodontic biofilms. And so we made the point that we're still dealing with polymicrobial situations where, just having a singular organism, that we can say, okay, this is the cause of, the infection we're seeing here, really cannot be done because, we're dealing with multiple organisms, and sometimes things may change, depending on, the environment, depending on, what types of bacteria the person might be exposed to when they go home, what they eat, what they drink, all of those different things, during the course of treatment. Those can, vary, from person to person. But then, one of the things that we want to kind of still remember, and we've looked at biofilm formation in different ways, I've given you different examples, we're just looking at, the same kind of thing. And again, the whole point of it is just understanding how we get colonized. And so we've seen different forms of this, including the slide that has, the multiple organisms within the biofilm, including the fusobacterium, that kind of bridges, the early colonizers and the late colonizers. And I made references to that yesterday a lot, so the whole point of it being that, please take a look at that one slide to remind yourselves of the organism, and that's why, I'm repeating it again today. But pretty much, what this is showing you, again, is the things we've talked about, where we do, the initial establishment of the biofilm, we have the colonizers, then we begin to increase the number of cells we find there, because, again, by quorum sensing, we can actually recruit cells, bring them into the community, and, as well, we can repel others, from the community. And so when we saw in the one image, we had the 2 points where you could have cells, essentially desorbed from the film, and by physical activity, cleaning, any kind of mechanical disturbance of the biofilm, we would have additional cells that could leave. And I pointed those two places out to you. And so again here, we're seeing again from the initial colonization, maturation of the biofilm, the secretion of the matrix material that kind of makes the cells aggregate and adhere to each other. And again, we're seeing the business of the individual cells that can detach, from the population. So again, we made a number of key things. We said at the center of the biofilm, right, we have, organisms that are principally what? Are they aerobic, facultative, strict anaerobes, right? Also the strict anaerobes, right, in there. And then we have facultative organisms, and then we can have, in the peripheries, we can have facultative aerobes, and some aerobes that show up, within there. So again, quorum sensing, is pretty important, right? We made mention of things about that yesterday. And again, this is pretty much just showing you this, what happens, when we have the organisms within, the biofilm, so we can begin to kind of aggregate ourselves in there. And so, once cells are growing, colonies are established, we have the maturation of the film, right? And we begin to see regulatory activity, within that biofilm. And those regulatory activities, underlying that is the quorum sensing that's still going on. And so we want to kind of remember that, it is both an ability to attract cells to the community, as well as being able to keep away, organisms as well. And so again, within these communities, we have organisms that are kind of interdependent on each other. And, as a result, of that, we can have, exchange of genetic material, which we talked about, right? So we can have plasmids, that are, moving about. We have transposons that can, also move from cell to cell, cell to cell. They can, transfer genes that are important for encoding molecules that can allow the bacteria to resist antibiotics and things like this. So we looked at that. So again, we want to kind of remember, what's going on, and other things that those organisms, might have as a result of those plasmids in there would be the ability to encode for things like toxins that can, be important, and other virulence factors that can make, pathogenicity, possible. And so again, as we look at what happens as infection progresses, right? So, we can have, within the pulp chamber and the root canal system, we can have necrosis, begin to take place, and this will be, a nice reservoir for infectious agents. The bacteria that are present in there can be now responsible for acute apical abscesses and cellulitis that can occur. And, things can still remain localized, even to a tooth or to neighboring teeth. But as we mentioned yesterday, if we don't have any intervention, when we begin to have such growth, we may have the cells, extend, outside of that local environment and begin to affect, the soft tissues. And so we have, a way of talking about these things. And so before we do that, let's just kind of remember what's going on over here. So when we first introduced the biofilm, we said we can have that, different locations, in the mouth. So we can describe, them, above the gum line below. We can have them on the tooth surface, for caries. We can have them in the cheeks, etc. So it all depends on where you have the bacteria in there. And so, we have for the film to form, right? We have that pellicle formation, the initial colonization. Okay, we begin to mature the biofilm, and then the various things we've talked about now, begin to occur. And so, when we look at biofilm, with endodontic situations, we can have, within the canal, intracanal, we can have extravascular, we can have periapical. If we have, and we mentioned this yesterday, if we have, any kind of implants, things like this, we'll give opportunity for cells to colonize those as well. And so, again, we can have these, foreign body-centered, type of infections, or biomaterial-centered type of things. And so, again, this is, just basically allowing us to get to the section on the abscesses. And I just, kind of put that in here. I was looking for something much simpler and better to show, but I couldn't find one. But in any case, it just kind of shows you where things, occur. And so we can have, abscesses occur. And so when we look at the case studies, later on, and we see the destruction, that takes place, on the face, as a result of this initial, lesions that start out, in many cases, as gingivitis. And when, people are questioned, especially in the case of the children, right, how long, has, this lesion been here? When did you first notice that something was really going on? And many of them will say, no, it was just a little kind of pimple-sized thing, that we noticed, in the mouth, and within a couple of days, you have this, massive destruction as a result of the bacterial growth and invasion. And so we can have localized, a collection of pus in alveolar bone at the root apex of the tooth. And this can, occur secondary to other things that are going on, manipulations in the mouth. And even, the situation with dental caries where, again, we don't have the huge involvement of the immune system, with the recognition of the bacteria and the inflammatory reactions that occur. But even that can be, the initiator, okay, of what ends up, with these abscesses. Other trauma, deep fillings that are done, and failed root canal. And so, the initial symptoms, pain, swelling, erythema, can begin to occur. And so, right here in this last one over here, if you're asked, okay, what's the reason for failed root canal? Okay, what would be the response?
Maybe like not proper removal of tissue?
Okay, not quite that.
Okay.
So what is it?
Incomplete removal of the bacteria?
Okay, yes, so not the tissue but the bacteria, right? So, bacterial infection, okay, is, really what you have as the reason for the failed root canal. Okay. So remember what we said yesterday, how you look at this as you would look at a wound. Everybody kind of remember saying that, right? So you kind of treat it that way. And so, the bacteria remain there, things are not, clean, you have those organisms there, you have them beginning to multiply. And so a large part of that also is the type of bacteria that you will have, in those situations. And these are anaerobic bacteria, okay, for the most part. And so the infection can spread, okay, from there, to the tissue of the face and the neck. And so some of the characterizations, as shown here from Shweta and Prakash, the publication, right? So they, add, in terms of the characterizations, hoarseness, drooling, okay, and in those cases, there might be fever, maybe no fever, and those tend to represent, very severe situations, emergency, because again, depending on what organisms you have, present in there, they're multiplying, fairly quickly, okay? And so you can have inflammatory responses because again, host immune system, is responding to antigens, and we have, many antigens, again, polymicrobial situation, and so, everything from facultative, to the obligate or strict anaerobic types. And in here, we also will still have, the presence of the virulence group streptococci. And so, so when we looked at caries and we mentioned, Streptococcus mutans, right, being very important and so on, and we don't think that, we could have them being significant here, but they can be. And so what you want to be able to remember, of course, is again that your strep, these are gram-positive organisms, they're facultatively anaerobic, right? And we made a differentiation between them and Staphylococcus. Everybody kind of remembers that. We'll review that a little bit later on. And so the key thing here with this group, the virulence group, Streptococci, right? So what is this referring to, the virulence group? What type are these? right? So we mentioned, the streptococcus, we mentioned that we have these classifications based on antigen. We talked, mentioned real briefly about the Langsfield grouping, right? We have the group A, B, etc. And we mentioned that we have the beta hemolytic strep. Everybody remember that on blood agar, right? The beta hemolytic strep. And then we mentioned those that are alpha hemolytic, right? And so alpha hemolytic versus beta hemolytic, what's the difference? What's the difference between those two? Beta hemolytic, we have complete hemolysis, right? So we have like strep pyogenes, right, which we looked at. And then, this viridins group, right, alpha hemolytic, so that's partial hemolysis, right, kind of greenish, color on the blood agar, okay, which is, where the viridins, comes from, right? And so these, mutants, streptococcus, fall in this category, okay? And so these are the ones that we typically associate, with commensal, features, within the oral microbiome, right? But we do have the fact that we can actually pinpoint strep, mutants, right, for, being one of the, the key organisms for, being able to metabolize, sucrose, fructose, etc., the acid end products that, we have associated with the, mineralization, demineralization cycle that takes place to lead to the caries. So they're important even here. And then we had the Prevotella species, Fusobacterium, and then, we're dealing with an anaerobic environment. Okay, so the Clostridium species, they feature prominently here. Because these are all, anaerobic, strict anaerobic organisms. And then, we have Treponema, we've encountered before. And so, again, in the studies, about 79% of the dental abscesses have, Treponema denticola represented there. And so there are different things that can occur. So again, typically, if, there's no infection, things can be fairly, cleaned, sealed, etc., and, nothing, happens, no infection. So again, when things are successful, as we already established, if we have a field, situation, we have bacteria. Okay, bacterial infection is present. And then, if that's not sufficient, okay, so, you need to look at other things that need to happen. And so, looking at the anatomy of the tooth, as we're going to see in some of the situations we have, that might actually give an idea as to what is going on. And sometimes, access, to the tooth, may be a problem. There may be, calcifications, etc., draining. Patient is so comfortable. And so again, you might have, repeat treatments, or in some cases surgery, and have improvements. And other things can be done, antimicrobial agents, to help to dissolve in necrotic tissue. Sodium hypochlorite is one that you see mentioned a lot in some of the investigations, that we mentioned yesterday, where people are actually looking at these lesions and actually trying to isolate organisms. You see them make mention of the use of this. EDTA is something else that's used, for demineralizing smear. Okay, that's caused by the instruments being used. Chlorhexidine, again, things like this can bind to the dentin and can allow for the antimicrobial, treatment, to be prolonged. And again, this is, known as substantivity. Other things can be used for dynamic energy, UV, light lasers, etc. Calcium hydroxide, the temporary filling sometimes, between appointments. And remember we mentioned yesterday that, individuals can have contamination occur in between treatments, right? When they go home, depending on what they eat and so on. So this is a way, to kind of, help to cover that, so that you don't have, the contamination take place. And so some of the organisms that, we encountered here, which we talked about yesterday, Porphyromonas species, etc., trichonematizole, gram-negative organisms with, the LPS, the endotoxin. And so, you want to be able to have that inactivated as well. And so all of this is, done to kind of allow the treatment to be successful, okay? Keeping, the numbers of bacteria not just down, but really being able to try to eliminate them in the focal site, that one is working in. And there's a lot of, interest, in the literature. If you put these terms in, you'll get all kinds of papers, people studying ways, to still have some success in this area. So nanoparticle research is pretty important. And so the nanoparticles, here have things like, chitosan or zinc oxide, in them. And, again, the whole idea being, those bacterial cell numbers, as much as possible, keep the numbers, very low, to being able to actually, exclude them from that focal site altogether. And so the treatments, done again, as we mentioned, similar to what you would do with wound infections. And again, to ensure, that the bacteria really don't get access into that site, right? This, meticulous attention that has to be, followed to allow this to take place. And so antibiotic use, right, is kind of associated with, the other treatments. And again, the idea is to keep the bacterial growth, away from those areas. And so we can have situations where we have complications and severity, of infection. And so again, Porphyromonas gingivalis, we've seen and talked about a lot, okay? And so, in abscess, abscesses that are formed, this organism is found in there. And so, remember what we said yesterday, right? So you may, isolate the organisms, from a number of individuals and find that not every single person has, P. gingivalis present, right? But it's, common enough in the isolations to where there's a lot of interest in it, okay? To make sure that, that's one organism that, that can be identified. And so in chronic conditions, as shown here, so, in autoimmune type disease, we mentioned diabetes, arthritis, right? We can have this, kind of tension between being able to really get rid of the organisms, being able to have, nice, clean, closed situation versus, this repeat visits because, you still have lesions, you still have bacteria that you have to deal with. And so in these conditions, you have peritonitis from, young aging individuals, you'll have, loss of teeth, and the population that you have, within the biofilms, right, can change. And so that's, another problem. But, the organisms that we've already identified, right, that, we've associated with, peritonitis are shown here. So we have Treponema denticola, we have Porphyromonas, Tannerella, right? You have Fusobacterium, etc. All of them, occurring together. And so if we have, both the availability of the bacteria, their antigens, whatever they're secreting, are present, and then we have the host immune system, responding, and we have inflammatory reactions. And if you remember what we talked about, that, organisms like Porphyromonas gingivalis can actually prevent the recruitment of, cells like neutrophils. And so, that whole combination of things, right, may actually end up making things worse, okay? And so, we kind of want to remember that part. So we have other conditions. So pemphigus vulgaris is another autoimmune type of condition that results in blistering. So you can have blistering that occurs, on the skin, in the mucosa, and in some cases can be, actually quite severe. And so when you have that, if you have in the gingival areas, you can have sloughing taking place, erythema, and desquamative gingivitis. So we have antibodies that are reacting to desmoglein 3 and also the type 4, type 8, type 7 collagen, laminin protein. And so, when you have these conditions occurring, right, there's an increased risk for having peritonitis. So we need to remember, we're not looking at the great big details of things, but we need to be able to remember the terms. Like, so we mentioned type 2 diabetes, we mentioned systemic lupus, we mentioned pemphigus vulgaris, and so on. And so we need to be able to recognize them and the association that they have with disease. And we mentioned also that this was a pretty much of a surprise to identify Helicobacter pylori in dental biofilms and saliva, because we typically think of Helicobacter pylori, the duodenum. But again, this has been recognized. You have several studies that have looked at this, and so the group of studies, have looked at dyspeptic children. And so you have, growth of these organisms farther up the GI than, you would expect. And so even, and then that oral cavity, they become part of the biofilms that we have formed over there. So something else to remember. And so when we have these organisms, within the oral cavity, right, we can have them, breach the epithelium. We can have them, in connective tissue, and we can have them in the blood vessels. And so when this happens, we have an increased risk for systemic disease. And there are, a number of different factors and risk factors that are associated with that. So, if you have situations, as we'll see, a few examples over here. So, preterm, low birth weight, babies, born due to bacterial and bacterial products being delivered into vascular circulation, we can have bacteria present, within the vascular system. Organisms that have been implicated, here's that Porphyromonas again, and Fusobacterium nucleatum. And, bacteria, and again, this is not unique to bacteria. We can have, bacteria, parasites, fungi, they can, perform, mimicry. And, what that just says is that the organisms either do one of two things. Either they express products, proteins, other molecules, etc., that look like host proteins. And these are coated on the surface of the organism. And essentially fools the immune system into thinking that those molecules, are self. The other way they can do this is to actually, recruit host molecules and coat themselves with that, and again fool the immune system into thinking that that's, that's just self. In which case, the host immune system doesn't, doesn't go into an attack. And so, when we have this, okay, a consequence that can occur, depending on what molecules, the pathogens have now put on themselves. We can induce autoimmune reactions. So, if the molecules, have portions on them, where you have epitopes that are dissimilar from what you would have normally, right? You can have an immune reaction generated against that portion, and consequently, you can have, antibodies and, activated cytotoxic T cells attack the host tissue. So, we want to look at some of the things that can happen as we get cells out of the oral cavity. And so, we already talked about the strep, the mutants group. Okay, they can use carbohydrate sources, etc. Produce the metabolic products, acid that can begin to affect, the enamel of the tooth. And so, as we have, various environmental features impacting the growth of those cells, right, we can begin to see changes occur. So we can have the strep in this case, be able to, as we already mentioned, reach the connective tissue layers, of the mucosa, get into the blood vessels, and we can now have them travel within, the vasculature. And places where they may end up, okay, would be, in the heart, around the heart valves. And when we have growth over there, we will begin to have tissue destruction, we begin to have clots form, and, you know, in addition to the bacteria themselves being present, you know, we may also have, you know, clots, you know, that can block the blood vessels. And so this is one way to get things out. So again, these conditions, pH, temperature, oxygen, you know, requirements and availability, you know, the inability of the host cells, you know, to produce molecules that can actually kill the bacteria before they get to these sites, right? All of these things can, you know, impact what the bacteria themselves are doing, okay, within these environments, and that will also affect, you know, gene expression in these cells. And so if you have strains that, you know, are able to produce virulent molecules and markers, then that would make them more pathogenic within the host. And so again, the mutants, strep, okay, are ones that we typically think about that can leave the, you know, the oral environment and get out into the tissues through the blood. And so when you have the bacteria, and we've already seen this, so we kind of, you know what's going on, right? We're going to have both innate and adaptive immune responses taking place. So in innate, you know, responses, right, we have antimicrobial peptides that are generated, that we have, you know, in the mouth, you know, we have things like lysozyme, etc. in the saliva. We, you know, the mucosal area, again, unless, you know, we have an invasive situation where we can breach it, that can actually provide a barrier. We have complement proteins that, you know, can be activated to, you know, the bacteria in innate system. So again, you know, that would be your lectin and your alternate complement pathways. And, you know, toll-like receptors binding to, you know, to camps on these cells. And so you have that generated, and if we have processing of the antigens and we now have them presented to T helper cells and to cytotoxic T cells and so on, if we have B cell activation, then we can have antibody production. We can also have, you know, these activated cytotoxic T cells also there. And so again, you know, from the oral environment, you know, draining into lymph nodes, we can have those reactions that we've talked about. And if the immune response, you know, is robust enough, we may be able to clear the infection. But most times, clearance of the infection needs both the immune response as well as intervention that's being, you know, done directly in the mouth to reduce the numbers of bacteria that we have in there. And so what we're looking at over here is, you know, streptococcal infective endocarditis. And again, you know, when we think of streptococcus leaving the oral cavity, getting into the tissues, right, we're thinking of, you know, mutants, but we have other species. So Streptococcus sanguinis is one that is also implicated in endocarditis that are caused by organisms getting into the blood. So you have, you know, a situation where you have an injured valve. You know, you have, you know, the cells, again, the oral cavity. And if they're able to exit the oral cavity, get into the blood system, right, we may find them, you know, in this environment. And so again, typically, if there is any kind of injury, we begin, you know, the clotting cascade, you know, to try to stem the flow of blood, and we form these clots. And so in the absence of bacteria, you know, this is aseptic. But then, if we have the Streptococcus sanguinis, we can, you know, have them, you know, get into the blood vessels, and we can have the induction of platelet activation. We bring in additional cells, monocytes, things like the tissue factor activity cytokines, and then, you know, we now have a situation where we're no longer aseptic, right? So we have bacteria present now, so we have, you know, a septic situation. So in terms of what you need to remember in this scheme is, you know, what we've just said. We go from a situation where we're aseptic, we have the injury. Normally, you know, we can have, you know, the clotting take place, have repair, you know, take place. But then if we have bacteria in the mix, then at that point, you know, we're going to now need to stimulate immune response. You know, we're going to have the reactivity, and if we don't have any intervention at that point, we can really begin to damage the tissue. And so that's where you know, a lot of the concern comes in, is that, you know, you now have, you know, pro-inflammatory cytokines being released. You know, we're bringing in cells, you're going to have more damage take place. And so we want to remember this species, okay, as different from, you know, the mutants. And so, okay, we're pretty much seeing the same thing. Okay, so you know, within the oral cavity, you know, plaque, you have the organisms present. We can have the components that the cells are producing, which we've already talked about, right? We have the LPS, you know, because the gram-negative cells, you know, for the most part. When we have that occur, right, we can react to the LPS. And so we can also have, you know, from within there, once we're in the tissues, the host immune response essentially mounts this response that is, you know, essentially systemic. So we're dealing with, you know, all different parts of the body at this point. But right at that tooth surface, you know, the things that we've already talked about, as we begin to get, you know, deeper pockets form, more organisms can get in there. You know, once in there, you know, we can begin to secrete, you know, the various substances, you know, that will lead to, you know, the inflammatory response that's generated. And as we begin to do that, you know, we increase the amount of cytokines, chemokines that are being produced, and then, you know, we're going to see this kind of, you know, widespread effects that we're going to have around the tissue. And so the cytokines, you know, which you're already familiar with, you know, IL-1, TNF, right, the chemokines, you know, IL-8, you know, all of these, you know, become really important. And so we see these features down here. So we have macrophages, you know, once they're recruited in, can take up lipids, and they will also be responding to signals. You know, when we have cytokines interact with them, things like IL-12, etc., they will respond by producing IL-1, TNF-alpha. We begin to see smooth muscle cell proliferation, you know, in the blood vessels. And so, you know, again, the intima, right, or the layer of the blood vessels most intimately in contact with, you know, the blood flowing in the lumen of the blood vessel. We begin to see proliferation of the smooth muscle, you know, within there. And then we begin to have C-reactive proteins, and they will, you know, essentially indicate the extent to which you now have this, you know, systemic inflammation that's occurring. And so we need to remember those things. And so we want to look at bacteria, in this case the gram-negative cells. So we mentioned features of Cochromonas gingivalis. We've talked about that, so we know, you know, what we expect to see in gram-negative cells. So everything from the LPS we've been talking about, the outer membrane that's there, the very thin layer of peptidoglycan, right? You know, we mentioned, you know, these cells, you know, you can either have them being motile or non-motile. They may have capsules or no capsules. So all the different things that we've talked about. And so just like we did for the gram-positive cells, where we looked at Staphylococcus aureus and looked at, you know, Streptococcus pyogenes, right? We want to look at, you know, sort of a sampling of gram-negative cells in here. And so we're looking at E. coli, and for the most part, okay, we have E. coli, you know, in the GI tract. You know, most times, you know, they don't do anything to cause any harm. But remember, we've been talking about strains and pathogenicity and virulence. Everybody kind of remembers that. So, which means we have E. coli strains that, you know, pretty much, you know, don't cause any problem, right? I mean, you know, we're concerned about their LPS because if they die, you know, we still have LPS that could be an issue. But we have E. coli strains that are pretty serious in terms of what they can do to the host. And so one of the things that we think about with E. coli, right, we associate it with traveler's diarrhea. But, you know, we have other organisms that cause traveler's diarrhea. We have, you know, protozoans, you know, we have these viruses, you know, that can make that. Typically, you know, when one travels and visits, you know, a different geographic area, within a day, or 2 days, the local flora, that the person is exposed to, might be the cause of, of this diarrhea. And again, the reaction of it is because this is something that, the person has not normally been exposed to. And in immune competent individuals, that might just run its course, within a few days to a week, the person feels okay. But in situations, where there might be other things going on, people are immunocompromised in any way, this can lead to more serious problems. And so E. coli, amongst the bacteria is pretty important in this kind of a thing. And so we have the strains that are recognized, 6 of them, we're going to look at them. And so we're looking at this image over here just to show what happens in terms of breaches to the epithelium. So we're looking at epithelial cells, the GI, columnar cells. And so if we look at the types, so we have the enterotoxigenic E. coli. And so, remember we talked about enterotoxins, we talked about the endotoxins versus exotoxins, right? Everybody kind of remembers this. This is one of the things that we're going to have to remember for the exam, right? If we mention endotoxin, we're thinking what kind of bacteria? Gram-negative, right? So when we mention endotoxin, right, what's the other name for endotoxin? What's the other name for endotoxin? I kind of hear it.
Anaerobic?
No.
Good today.
Okay, so what's the other name for endotoxin?
Good today.
Okay, well that's the part of it. We won the whole thing. The LPS itself, right? So the lipid is the active component of the LPS, right? So that's the endotoxin. But then remember what we said. We typically associate the exotoxins with gram-positive cells, right? But we do have exotoxins that are produced by gram-negative cells. Okay? So it's not the first thing you're going to think about, but they do produce them. And so we're seeing here an example of that situation. So we have the exotoxins, okay, that are responsible for the diarrhea. And the other thing we mentioned with endotoxin and exotoxin is that exotoxins are typically proteinaceous, right, compared to the endotoxin that has the lipid A component in it. And so again, we're distinguishing these enterotoxins from the endotoxins by their heat lability. Stability. Okay, then we have the enteroinvasive E. coli. So these we can have them within the intestinal epithelial cells. They also can produce, the exotoxins, in this case a cytotoxin, and an enterotoxin that can, be important for the diarrhea. We also have enteropathogenic E. coli. And so these ones can cause, effacing lesions, and they're basically, destroying the brush border of the intestinal epithelial cells. And again, when they do this, we can see, this destruction that takes place. We have the enterohemorrhagic E. coli, also producing effacing lesions. But then here we're, looking at hemorrhagic type situations. So the inflammatory reaction that's being produced, has within this, hemorrhaging aspect of things. And so we also have the production of toxins like we see in Shigella, okay? We have the interaggregative E. coli, and so these, would adhere to the epithelial cells, kind of clumping up against them. And so we have the term, a stacked brick appearance, to refer to these kind of clumps that occur with this particular strain. And so unlike the other ones, we don't have, a particular toxin that's been associated, with this. And then we have the diffusely adherent E. coli. And so again, the adherent, the pattern that's being formed is different from what we have here. And so again, if we have situations, where we have malnourishment, if we have any kind of, immunocompromised type of states, in particular in children, this can be a problem with this particular strain. And so a lot of things that we've talked about also feature here. So again, if it's traveler's diarrhea, people are usually questioned about history. Depending on how severe things are, there might be a need to isolate the organisms, from fecal samples and try to identify it. So everything from serology to molecular probes can be used. Okay, so the same things that we've talked about. And in these cases, if we have symptoms, and again, if they're quite, a serious antibiotic treatment, may be given, for that. And again, contamination of food and water is usually the source. So again, avoiding those, and that's also important when a person actually has traveled to a different location. For those first, few days to a week, it really kind of makes sense to, avoid the sources that might actually have the bacteria in them. And usually after about a week or two, most people are kind of acclimatized to, the organisms that you have in that new environment. And so we can have, sepsis occur, and, when that's occurring, we can see the different, features, heart rate changes, respiratory rate, blood cell count, with the leukocytes. And, if things get pretty severe, there might be septic shock, extreme drop in blood pressure. And so again here, it could be a combination of not just gram-negative cells like E. coli, but you can have other bacteria as well. And depending on what's going on, this may be from fungi, and even protozoan infections can kind of give the same thing. So, and this is why isolating and being able to identify becomes important. Salmonella is another important one. So every few weeks you hear about salmonella, contaminating, foods and so on. And so we have, quite a large number of serovars, more than 2,000 salmonella serovars, the gram-negative, non-spore forming. And so amongst the group of enteric organisms, E. coli, Salmonella, Shigella, all Vibrio, all examples of that. And so contaminated food, everywhere from beef products, poultry, eggs, so on, we'll have the shellfish, for Vibrio, we will have these gram-negative organisms. And so when we have them within the GI tract, right, we'll have them producing these cytotoxins and enterotoxins that are there. And so abdominal pain, cramps, diarrhea, vomiting, fever, these are all associated with these kind of infections. And so again, being able to isolate the organisms, from stool samples, kind of verify what they are is important. And, mainly the identification serves one very important purpose, right? So because you have so many different strains, and these strains can be geographic, it always becomes important to know what serovar you're dealing with. And so that's where things become important to be able to do that. And so because of the extent of fluid loss that occurs, right, being able to replace electrolytes and fluids and all of that become important. And so again, prevention, good food handling practices and so on, adequate cooking, cooking things well done. And most importantly, if a person, has an infection, they should not be handling food. Okay, they should not be, preparing food, serving food, any of those kind of things, because the potential for, spreading the infection to others is very high. And so we have typhoid fever, and again here we have, Salmonella enterica subspecies with the serovar typhi, and this is a gram-negative rod, and again from food contamination. We have a mild form of infection caused, parathyroid typhoid fever. And this particular organism and the infection caused has a very important place in the history of microbiology. And so, whenever, we have a course like this, it's always important to kind of remind ourselves of those things. Very much like we talk about, Koch's postulates and everything, we kind of introduce that at the very beginning, because we want to know the historical association of that with how we think about infection. And so in this particular case, bless you, we have bacteria that can leave the small intestine and they can travel, you know, into the lymphatic tissue, blood, liver, gallbladder, so extra-intestinal, and they can, you know, affect other parts of the body. And so individuals, you know, will have things like fever, headache, you know, abdominal pain, depending on how severe, you know, they may become anorexic and all of that. And so individuals can be carriers and not show symptoms. In other words, they're asymptomatic carriers of Salmonella typhi. And so again, you know, the historical connection, which is, you know, why we're doing this, besides just wanting to know about the gram-negative cells and how they cause infection, is that, you know, we have typhoid Mary, and in microbiology history, this was an individual who worked in the food industry in many different capacities, both, you know, commercially and in private, and she was a carrier. And so these organisms, you know, they can grow in the gallbladder, and, you know, in the carriers, you have the organisms there, but they themselves, you know, the carriers, are not showing any symptoms. And so, because they can contaminate, you know, hands and so on, they can contaminate, you know, food, you know, during preparation, and then individuals who were susceptible now, you know, would get sick, and in some cases would die. And so, once it was clear that she was contributing to these infections, you know, she was asked to stop working, you know, around food. You know, she would stop for a while, and then she would show up working somewhere, and all of a sudden, you know, cases would flare up, and then, you know, it would be clear that she was working again. And this happened over and over again. You know, she was, you know, she stopped working commercially. She worked for private people, you know, cooking for them and so on. And again, cases would come up, and it would be clear she was working again. And all the whole things that we think about in public health, you know, where you quarantine people, you try to keep them away from, you know, everything to protect themselves and everybody else. You know, she managed to, you know, escape all of that. And, you know, so, you know, in the history of microbiology, we have that situation, and she was, I think she worked in New York, I think is what it was. And so we have this situation, you know, where very clear demonstration of a person who is a carrier, contaminating food, and essentially making, you know, the public ill as a result of this. And so, you know, we always, you know, kind of look at that to remind ourselves, so that even today, when people, you know, have, you know, things like, you know, norovirus, you know, giardia, or in the case, you know, Salmonella typhi, you know, if you're sick, don't go to work, right? If you're, you know, known that you're a carrier, then, you know, don't work in that industry, because the potential of making people sick is so high, you know. So we want to remember typhoid Mary and Salmonella typhi. But in any case, being able to see the bacteria in, you know, blood, urine, or stool, being able to use antibodies to identify, you know, the cells, antibiotics, you know, is used, you know, with vaccines for high-risk individuals, and the thing of, you know, purifying the drinking water, preventing food handling by carriers, you know, being able to isolate, you know, patients that are infected, because the, you know, the potential to get the contamination to other people is very, very high, you know, due to the contamination by fecal material. And so again, you know, the particular serology, you know, the test, you know, that's used to look for these things is known as the Wido test. And so we have Shigella, which, you know, you don't hear about very much, but many times, you know, again, depending on what location you are, if you have Salmonella infections, you know, and so on, you know, invariably you're going to hear about people also having Shigella infections. These are gram-negative organisms, again, just like the Salmonella, right? We have, you know, 4 species of Shigella that are important. You know, we just want to know just the genus. They're intracellular parasites, that's a key thing, a difference here. And, you know, they're within, you know, the epithelium of the colon. And again, we can have, you know, inflammatory reactions in the mucosa, and, you know, humans, you know, the only known host, so we don't have a situation where we can get contamination from animals and so on. So they're important in that case. So we have endotoxin, right, from the LPS. We also have exotoxins. And so the Shiga toxin, you know, is one that, you know, you read about and hear about. And we have the E. coli that produce, you know, a similar kind of a thing. And so the important thing here is we can have, you know, the glomerular epithelium that can be targeted by this thing. And so, you know, we can have kidney failure as a result of this. And so again, you know, you have these things inside the cells. They have the virulence, you know, molecules that they're producing. and they're able to get those, into those host cells, within the epithelium. And so we have, watery stools, with blood and mucus and pus, and, serology, the various biochemical tests, fermentation tests, and all of these things they use to identify. And so antibody treatment, is given when things are quite severe, and then we can also have preventative things, okay, good personal hygiene and preventing contaminated water and so on. So, just to kind of help us, kind of remember these things. And so what we want to do, okay, we'll take a break, and then we're going to come back and we're going to look at the cases. So let me just pull that up while you're doing that. Okay, so we'll take a little break, 5 minutes, and then we're going to come back and we're going to look at these cases. And again, remember what we said. So, we're taking the approach where we wanted to just kind of see, what happens in these cases that start out with some of the things that we've already talked about, where you might have, gingivitis, where, under normal situations, intervention with that might actually help clear things up. But, in these particular cases, we see that things can extend beyond, just the initial case. And so we'll look at that, and then we'll look at pneuma, and we'll look at Pathogenicity, and see what happens in those cases. Just kind of help illustrate some of the things that we've been talking about. So we'll take a break, and then we'll come back and start looking. This section, again, remember what we're trying to do, because of the nature of the course, how short it is, and so on, we're not having you read, these cases and, have them discussed and all of that, but we want to be able to highlight some things and, to kind of help us remember, how the association, between gingivitis, periodontitis, we've already looked at, how these can become associated, with life-threatening type of infections that can occur. And so, we're looking at two different types of things. So, we're going to look at a situation where we have a condition that's fairly rare, but depending on where you are, you might see, individuals with this, a little more frequently than other places. And so, we have the one situation, with Pathogenicity syndrome, or PLS, where we have a rare autosomal recessive type of a condition. And on the other side, we're looking at a condition that's associated with severe poverty in some cases. So in parts of the world where this occurs, the association with having pneuma develop is associated with children in very malnourished type states. Where they will contract this. Other situations that can predispose, individuals that have other conditions, immunosuppressed states, like things like having HIV infections and so on. And all of these together, combined, have this presentation where individuals essentially start out by having, this what could be considered, gingivitis, but then it becomes, something much larger, to where we have destruction taking place now in the face. And so the first one, that we're going to look at, okay, is condition from Pathogenicity syndrome. And so in this particular case study, where you had, an individual that essentially presented, with the condition that we're seeing over here, with, you had loss of teeth, extreme, erythema, redness in the gums, bleeding upon probing, is exemplified, by this. And so this particular condition, is not only something that's affecting the teeth, but also affects the skin, affects, the skin in different parts of the body, including, the palms of the hands, soles of the feet, that kind of a thing. And so the characterization of Pathogenicity is just this. Individuals begin to lose their teeth at a very young age. So when they have, the deciduous teeth, you begin to have exfoliation of the teeth, and you get to the point where the alveolar bone is affected, and you don't really have a good anchor for the teeth, anymore in the mouth, and they're mobile. And by the time it gets to that point, the teeth, will now be lost. And then there will be a period, of, if you will, like a rest in between, the point where when they start having the mature teeth, the permanent teeth form. And then the whole process begins all over again. So that by, the age of things like 15, 16, teenage years, even the permanent teeth, beginning to be lost. And so the important thing about it, which is why we're looking at it, is that it is the thinking is that it starts with periodontal disease. It starts with that. You have the gingivitis, that can also be present, and with those two things happening, right, we get to this point where we're now having the floating teeth that are occurring. And so, a couple of important things, good to note. And so when we look at this first image, right, so again what we see, is the effect that we have, in the gingiva. And part of what we've talked about with gingivitis and periodontal disease is the involvement of the host immune system. Okay, as everybody can remember that, we looked at the slide a couple of times, the interaction between, the antigens of the organisms, the response, innate and adaptive, and if, we still have the antigens present, we just have this continuous process, that takes place. And so that is going on here. Now what's unusual is that period in between, the loss of the deciduous teeth and when we begin to have the permanent teeth erupt and then have them go through that same cycle again. Another thing that's of interest to note here is this exfoliation that occurs on the teeth. So that part of it, it's not really clear why that happens, but it happens to the deciduous teeth. It also happens to the permanent teeth, both things. And so when you look at the other areas that individuals can be affected with in here, we've seen an example over here of the what happens to the skin. You have scaly lesions that can occur. You have this hyperkeratotic state that occurs. Not just the feet, the hands as well. And in some cases, it seems to be bilateral in what happens, in both cases. And then at the bottom of the feet, these yellowish bumpy structures occur. So that plus what's going on in the mouth kind of go together in individuals. Now there's some indication that it might be familial. So there are cases, in the literature where you have, family members, amongst the children, each one of them essentially has the same thing going on. And so, for most of these, antibiotic treatment seems to lessen the effects of it. And so when we have a situation like that, where if you're treating with antibiotics and you have, some resolution, then, the bacterial etiology becomes, pretty important. So, with that, you can say, okay, bacterial involvement, is important because, you're seeing resolution as a result of having the antibiotic treatments that are given. So for this particular condition, the things that we want to remember, the periodontal involvement in this condition, the loss, of the teeth, in both, the deciduous teeth, permanent teeth, and we also want to associate it, with the autosomal recessive nature of the condition, and then as well, the whole aspect of being a disorder of keratinization. So these end up being, important features of this particular condition. And so if you see those, you want to be able to correctly identify it as this, Papillon-Lefebvre syndrome. The next one we want to look at is pneuma. And in this particular case, we're looking at two types. So we're looking at, pneuma in children, the way it would be normally characterized, And then we also are going to look at an example of a case, that occurred in an adult. And so, we're basically highlighting the features of the conditions such that if you were to see the characterization, of the condition, you could correctly name it as such, okay? And so, we're going to look at the first instance, and of all the three case studies, that we're looking at, this particular one is one that I would say, at some point before the exam, if you can look at the actual case study itself. So, I don't know if you can maybe pull it up and have it available with the notes and everything. So that's the only one that I want us to look at all together. And so we want to kind of see the features that characterize what's going on. So this is a case of a child that contracted pneumonia, and 6-year-old had HIV positive and had malnourishment, and so he wasn't receiving his heart treatment for the virus, and so in addition to having the virus, this condition itself started. And so the point of it was that, again, a small lesion, kind of like a pimple-looking thing started, and within a few days, this bacteria were multiplying in such amounts that, this is the kind of a thing that occurred. And so what we're looking at here in the images, examples of where treatment had already started, okay? So treatment had already started in this case, and so we're looking at 2 weeks after admission of the child to the hospital. And so what essentially begins is, again, we have some of the same organisms that we've encountered, right? The strep organisms, lactobacillus, Potteramonas, all the different things we mentioned, Prevotella, Propiobacterium, that have been isolated from lesions in these individuals. The question is, what initiates it, right? And how does it start? And so one of the things that you will see being referred to for this condition is that we have a very aggressive necrotizing gingivitis that occurs. And so once that begins, very quickly, we can have these lesions that we're talking about. Now, we have diseases that are classified as neglected diseases. Anybody heard that term before? Right? And so this is one that falls under that category, okay, of neglected diseases. And this is because, in the areas where it shows up, there's a lot of poverty and so on. And most times individuals may not seek treatment right away. And so there's, whenever they get seen, the interest is to try to find out, what organisms can be isolated, to try to build a profile, okay, really globally, to find out, what are the core organisms that are found in these type of situations. And so the necrotizing gingivitis, is also found in other situations. So remember we mentioned, when we talked about periodontal disease, we mentioned, risk factor, smoking, etc. That can also be a contributing factor, because of the poor upkeep of the mouth. And so those conditions are referred to as trench mouth or meth mouth. And so trench mouth, was first identified, in soldiers in trenches and things like this, with the poor oral upkeep and, development of gingivitis, the mouth, and so on. And because of the polymicrobial nature of the infection, right, the smell associated with all that bacteria, right? So we have the term trench mouth for that. And so in meth users, that's also something that's seen. And so it can progress, to what we're going to see in the second case. And so those two terms, kind of refer to this. But they're separate from pneuma because the extension of that involves now the disfiguring of the face, as we see. And so with that intervention, this is what can occur. The other thing to kind of mention is that, the viral association. So again, here we have a 6-year-old HIV positive, right, malnourished, And then we have this progression that occurred from this initial, gingivitis that kind of spread. So other viruses that have been associated include, things like herpes virus infection that kind of initiates this. And, what seems to be very important in all the cases is this malnourishment. Okay, the malnourishment seems to be a very big key for that. And so again, we see, in here, the necrotizing gingivitis that occurs, necrotizing, let me get to the slide over here, sorry about that. We get to the necrotizing periodontitis. We have, as we begin to get into the tissues, we also can see, stomatitis occurring. And again, the key is that, these are polybacterial infections, and where you can isolate, different organisms, from these lesions. And so what's not really clear is the relationship between the bacteria, the immune system, right, and then the initiator, whatever that virus, infection happened to be, okay? And central amongst all of these is the malnourished state of the individual. So we need to just remember, the condition, be able to associate it with the things that we've talked about. So if we look over here, so this is a way of trying to understand mechanism. So, as we said, what's really going on? What's the immune system doing? What are the bacteria doing? And, what's the role that we have with the malnourished state and the immune suppression that's there? And so you see on the list over there, top, of the predisposing factors, right? We have malnutrition. But then we can have other things, viral situations, chickenpox, malaria, measles, and in the case that we're looking at over here, the HIV/AIDS infection. And so, with these occurring, and so in many of these, for example, in malaria, you have, cytokine storms that are formed as a result of having the parasite infection. And so you have this, dysregulated immune response that's taking place to, what you have as the initial organisms, of the infection, and then the bacteria that are now multiplying. And so we kind of compound that by having more cytokines being released. And so, with that, okay, you're going to begin to see, the effects, and the impact on the gingivitis that's already there, the periodontal, disease that's already there, and the stomatitis that's already there. And so in trying to understand what's going on, right, because there's so many question marks, right? So again, once you have the extent of the infection, right, what else are you seeing? So we have, teeth exfoliation, we have a lot of gangrenous tissues. So, in this particular case, of the 6-year-old, the initial presentation, to the hospital, if you saw that, you would think there was no resolution at all possible, but because of how bad the lesions looked. And so, so you have all the gangrenous tissue, the greenish appearance, that you have, of the tissue and so on, right? Because then, now you're in the soft tissues. And then you have the, mid-face destruction that can occur. So in some cases, aspects of the nose are gone, things of that nature. Essentially, you have holes in the face and all of that. And so, the bacteria, we can, have them, into the blood, so you can have septicemia, right? You can have individuals, severely dehydrated. And the interesting and kind of unusual thing about this is how much it doesn't affect the other parts of the body. Okay, so so even in immune competent individuals, as we're going to see in the next adult example, right? In that case, what seemed to be most prominent was that malnutrition again. So, and if there is no intervention, right? You can actually have them occur. So you have a number of features that are all interconnected in this, to have the bacteria present and to have the continuation of the infection that takes place. But then, as we could see, in these images over here, once things had been cleaned out, the sloughing of the skin, exfoliation taking place, right? They we're beginning to see, some resolution to, to the tissues. Now, in this next one, this next case occurred in an urban environment in Atlanta. And so, again, we have, acute pneuma with the necrotizing, ulcerative gingivitis, that kind of started things. And so, in this particular case, we're looking at an adult situation as opposed to the child situation. And so, some of the same common things, that you would see in the cases, is the necrosis that essentially extends, in a wide area of the face. And so, we already mentioned the terms trench mouth, meth mouth, you want to remember those. And so in this particular case, an individual, immune competent individual, is the one that showed up, with this case. And again, we're pretty much interested, in the features that we're going to highlight. And so, when upon admission, a lot of different things, respiratory distress, going on, acute kidney problems, many other things going on. But amongst all of that, was a very malnourished individual. And so one of the first things, that was done, was to look at, the blood culture to see what kind of organisms might be present in here. So you notice the loss of the teeth. Some of the teeth were actually removed, to prevent aspiration. But organisms, such as Streptococcus viridans, okay, so what are the viridans group again? Streptococcus viridans, the alpha hemolytic strep, okay, found. Staphylococcus epidermidis, okay, so we looked at that one to differentiate it from Staph aureus. Everybody kind of remember that? So if we're coagulase positive, then we Staph aureus or epidermidis, right? So that means we have to remember those things. So Staphylococcus, again, coagulase positive, and the catalase and so on. And so we had those there, but then there were additional organisms. Enterobacter aerogenes, okay, was also isolated, and Citrobacter fusari. And so, following the stay, right, so other things started to happen, hemorrhaging, in the individual, all these eschars that had formed, those had to be removed. And what was important was that it appeared that the facial reconstruction could be done, okay, as we see happen even in the childhood cases. But, unfortunately, after he was released into a skilled nursing facility, he passed away. So that reconstruction, did not take place. So for us, the things that we're most interested in understanding with these conditions is, first of all, that we can progress, from, gingivitis, okay, that we would think of as the thing that can be easily managed, or from periodontal disease, okay, a little bit more, intense in terms of the immune system response and so on, but that we can actually get it progressed, to what we've seen. And so, we want to be able to recognize those. We have questions about them. Be able to recognize the common terminology that's there for describing these. And like I said, of all the three cases, the one involving the child is the one that we can pull that up, and have that available, so that we can look through it in terms of the mechanisms that we talked about, and the understanding that's there in terms of what's known about organisms involved. So a question that always comes up, with a condition that we've just seen with all that bacteria and the destruction, is that is this something that's contagious? And and in reality, it's not. So in other words, being around a person who has that, or working with them or treating them, doesn't pose contagion. But we have all that bacteria that's there. And so in all of the different situations we've looked at, right, what's common amongst all of them is this polymicrobial nature of the infections, right? but we can identify, like we did with the periodontal disease, we can identify, key groups of organisms, and indeed we can actually mention a singular organism, not because that's the main etiology of the condition, but one that's pretty prominent in cases that we've seen for Papillomavirus gingivalis. So, so in terms of, the things that you need to remember with these for the exam, like the mechanism, the questions there, how we get to that point, with the necrotizing gingivitis, etc., being able to recognize that and being able to differentiate it from the actual condition, that is pneuma itself. Okay. So, so in the remaining time, we just kind of want to review a little bit, and just to mention some of the things, because it's been, 4 weeks, right? And you have all these other things that you're doing, so whatever we talk about the first week, probably, it's gone somewhere. We're not remembering it. And that's okay. You guys are so busy. But in any case, so we want to remember the general things, right? We still need to know, what the gram stain reaction is. If you see an image, can you tell? We need to be able to put together, pH conditions, oxygen conditions, for the cells, in terms of the gram positivity or negativity, to be able to isolate, to be able to identify organisms. We've looked at organisms that are strict anaerobes, facultative anaerobes. We've mentioned, strict aerobes like we have with the bacillus and so on. So we want to be able to kind of put them together. We looked at growth conditions, of these cells, so that we still need to know when something is, mesophilic, hemophilic, etc. And we still need to recognize those kind of things. And then we started to look at where we find these organisms, right? We talked about growth media, how we grow the cells, solid media, semi-solid, etc. And looked at what we need, for prevention, antimicrobial type things. And we stressed the business of being able to kill the spores, okay, in spore forms, right? And so the spores contain everything that's going to be required for vegetative growth. And so if we have spores around, right, yes, they're dormant, features of the bacteria, but if you have, conditions that are favorable, right, then we have vegetative cells that just grow again. So the idea is to be able to kill the spore. And so we mentioned, that, standardly, we want to be able to autoclave, things, 15 minutes, 15 pounds of pressure, and all of that, with the moisture, to be able to 121 degrees, Celsius, to be able to kill those spores. And then we, mentioned the fact that spore formers themselves are used as the indicators for whether you had, failed autoclave reactions or whether you had one that was, successful. Okay, so those kind of things. And enumerating the cells, counting the cells, went all over that. Then we started looking at the conditions. So again, with the carious lesions we talked about, that white spot that begins is the initial thing, right? That tells us that we have this condition beginning. And then, to remember the organisms associated with that, right? And how these organisms differ from the ones we see in periodontal disease, right? And we looked at that red complex, right? So what are the red complex organisms? Okay, I heard two of them. Okay, what are they again? First of the red complex? Okay, and another. Okay, so we want to remember that. So we know we have the other organisms there, right? I always want to make sure I say that, because when you look at it, the studies showed, right, that you could group things into, the orange complex, the yellow complex, the green, the purple, right, all of those different things, right? We had, in the green, these strep organisms there, but the three, okay, in that red complex, right, pretty important because, many times they're co-isolated together, okay? And so we wanted to remember that. And so, we looked at the different disease types. So again, look at those, to be able to differentiate them. And, when, again, looked at the group of organisms, we associate, with the endodontic lesions, right? How different are they from those that we see in periodontal situations, right? We want to be able to differentiate those. And again, the exam is all multiple choice, So which means you're going to either have direct questions or you're going to have scenarios, okay? And so the questions I'm going to send to you so that you can post, right? You can use those to review, okay? And so, so basically, those questions directly and material associated with them, okay? You want to go through so that, when you have the questions, you can easily recognize what you need to recognize and be able to answer the questions, right? And so if there are any questions, okay, as you're thinking about things and when you have the question set come in, please send questions to me so that way I can get responses back to you. So I'm going to do that as soon as I get back. That way you have, the rest of today, tomorrow, etc. going through. And so one of the things, with the questions, so because we've mentioned so many different organisms, right? As you're reading the questions, kind of pay attention so that we don't confuse, the different organisms. So they may be similar location, doing similar things, but one is gram positive, one is gram negative, one is strict anaerobe, another one might be facultative, so we don't get stuff mixed up with those. Okay. All right. Any questions? Okay, so I'm going to get that to you as soon as I get back in there. Okay? Okay. Well, good luck on the exam.